السلام عليكم ورحمة الله وبركاته. بدءا الشكر الجزيل إلى جامعة البصرة أتاحتهم لنا الفرصة في المشاركة بمؤتمرهم للعلوم التطبيقية. بحثنا بعنوان Prevalence of Antibiotic Resistance and Extended Spectrum Beta-lactamase in Proteus species isolated from patients with the urinary tract infection in Al-Najaf Al-Ashraf Hospital. Your speaker Dr. Thakra Abdullah Mayahi, كلية الطب جامعة الكوفة. أهلا وسهلا بكم. بسم الله الرحمن الرحيم. وعلمك ما لم تكن تعلم وكان فضل الله عليك عظيم. صدق الله العلي العظيم. Introduction of our study, urinary tract infection is among the most common bacterial infection both in the hospitals and in community. Urinary tract infections are defined as the significant bacteria urine in the setting of symptoms of cystitis or pyelonephritis. Most urinary tract infections are caused by the gram-negative bacteria, being Escherichia coli, Klebsiella pneumonia, and Proteus mirabilis, the most commonly isolated. The hallmark of the urinary tract infections caused by the Proteus species is their ascending character and thus predilection from the kidney, causing cystitis, pyelonephritis, and productions of urinary stones, which are a hallmark of infection with this organism. In my hypothesis in this study, in Iraq, Proteus mirabilis constitutes that's in third most commonly isolated pathogens of urinary tract infection after Escherichia coli and Klebsiella pneumonia. However, multiple drug resistance in Proteus species isolates, including resistance to an extended spectrum beta-lactamase, is arising, causing a growing public health problem worldwide. In Al-Najaf Al-Ashraf, there have been a few studies published on the prevalence of extended spectrum beta-lactamase producing a Proteus species isolated, and it is associated with the blacenteins gene. The aims of the study was to investigate that is in the occurrence of extended spectrum beta-lactamase and presence of a blacenteins gene among Proteus species isolated recovered from the patients with a significant bacteriuria. Objective of study. The present study is carried out to achieve the following objective: to determine the frequency of bacterial uropathogenic in urine specimens obtained from the patients with a significant bacteriuria, to in phenotyping investigation the occurrence of the antibiotic resistance and extended spectrum beta-lactamase of production among Proteus species isolate, to evaluate that in disseminations of blacenteins genes among isolate. Method of study. Cross-sectional study was conducted in 3-man hospital in Al-Najaf Al-Ashraf from the March 2092 in 2020 in patients who had clinical suspicions of urinary tract infection. First step, collections of urine specimens from the patients suspected with the urinary tract infection, then cultured on a blood and meconic agar plate, then identifications of bacterial isolation dependence on biochemical test, and beta vitamin system. When found a Proteus species, and then investigation, that's antibiotic resistance in extended spectrum beta-lactamase production, and detections of plant CTM genes by the PCR assay. End result found in this figure patterns of uropathogenic isolate from the patients with the significant bacteriuria in 1,132 cases, found about the Proteus species 6.5%. In this figure show Proteus about binary 1.3% and Proteus vulgaris 1.3%. A frequency of a Proteus species isolated from patients with the significant bacteria. In this table show antibiotic susceptibility expressed by the Proteus species isolated presented a reducing susceptibility to the quinolones. Number of cases 32. Found resistance ampicillin, amoxicillin, piperacillin, amoxicillin clavate acid, and the Thakra clavaric acid. In this table show distributions of multi-drug resistance and extensive drug resistance among 42 and Proteus isolate cases. Found 3 patients of multi-drug resistance and 12 cases extensive drug resistance. In this table show distributions of LAMP-CTAM's gene encoding gene phenotyping positive extensive spectrum beta-lactamase procedures among Proteus species isolation found in Proteus mirabilis about in 39 cases, found BLA-CTM's genes positive isolated about in 15 cases. In this figure show or image show the confirmatory test for detections of extended spectrum beta-lactamase productions phenotype by the disk approximation testing. Note that expansions of the zoom around that astronym disk adjacent to that amoxicillin's clavaric acid 22 and 10 mg showing the extensive spectrum beta-lactamase reducing by Proteus mirabilis isolated. This isolated harbors beta-lactamase CTM gene. And in this image show the ethodium bromide stained agarose gels of the mono-black PCR amplified product from the extracted DNA of a Proteus species isolation and amplified amplified with the placental gene primers. According to the results carried out in this study, these following conclusions have been reached at the Proteus species isolated are the further agents responsible for urinary tract infection in Al-Najaf al-Ashraf. 2. The present study highlights in alarming levels of the antibiotic resistance, multiple drug, and extensive drug resistance among isolation. The results of the study reflected the inconsiderable frequency of extends spectrum beta-lactamase among the isolation in Najaf al-Ashraf hospitals. For molecular detection revealed as a relative high frequency of the placental genes in the Proteus species urinary tract.